What is an action potential? What is synaptic transmission and how do they work? Well, an action potential is simply an electrical impulse in a neuron that is caused by a stimulus. In this picture, the stimulus is a thought. It's hard to see, but they're thinking, what is the powerhouse of the cell again? Hopefully you know the answer to that one. Anyways, before the action potential, there is the resting potential, where sodium channels and potassium channels are closed. These are the sodium channels, these are the potassium channels. The sodium ions are on the outside of the cell, and the potassium ions are in the inside of the cell, as well as negatively charged proteins and polyatomic ions, like that protein is. This causes there to be a membrane potential of negative 70 millivolts, which can be represented on this graph here like that. That is the resting potential. Then a stimulus, like the thought, causes some of the sodium channels to open. This causes the membrane potential to slowly rise until the threshold is reached, which is negative 55 millivolts, which can be represented on the graph also. And that is the threshold. And then once the threshold is reached, the voltage gated sodium channels open. Like this is a voltage gated sodium channel. They open, and this allows the sodium ions to flow down their concentration gradient into the cell. This increases the membrane potential, which causes more and more voltage gated sodium channels to open in a chain reaction. And this causes a rapid increase in membrane potential, which reaches about 30, positive 30 millivolts. This stage is called depolarization and is the actual electrical impulse that travels down the axon. The next stage is repolarization, where the sodium channels are closed, as you can see, and the potassium channels are open, which is signaled by the positive 30 millivolt membrane potential. This is the sodium channel, they're open. The potassium ions also flow down their concentration gradient from the inside of the cell to the outside, which is the opposite of sodium. This lowers the membrane potential so that the neuron can get back to the resting potential, but the potassium permeability stays high for a bit longer, causing the membrane potential to dip below the resting potential to negative 94 millivolts, which is called hyperpolarization. So on the graph, this would be repolarization, but then it goes below to negative 94, which is hyperpolarization. And then to restore the membrane potential back to negative 70 millivolts. Okay, and then finally, the potassium channels close, and the sodium potassium pump starts leveling out the sodium and potassium to restore that resting potential, which is right here. This is the sodium potassium pump, and then it's restoring the balance. Okay, so the action potential traveled from the cell body down the myelinated axon to the axon terminals, and at these axon terminals, like these, that's where synaptic transmission occurs. This here is the synapse, and it's simply a junction between two neurons. The action potential, which was represented by this like blue electricity, triggers the opening of these calcium channels, to which calcium ions flow into the presynaptic neuron. The calcium ions then activate the synaptic vesicles, which are these red things, inside which are inside of the synaptic knobs of the axon terminal, which are they're filled with neurotransmitters. The vesicles, through exocytosis, merge with the presynaptic membrane, which would be this, and release the neurotransmitters into this space, this right here, which is called the synaptic cleft. The neurotransmitters travel to the postsynaptic neuron, which is this neuron, which has receptors for the neurotransmitter on its surface, which is represented by these like red things. And the neurotransmitters will bind to it, and that will cause sodium channels, like this one, to open, which signals another action potential so that the message can continue going. The next thing they'll do is the neurotransmitter will unbind, and the autoreceptors on the presynaptic neuron will regulate the concentration of neurotransmitters in the synaptic cleft through a negative feedback loop. There are two paths. One is the reuptake transporters, which would be like right here, take back the neurotransmitters into the presynaptic neuron, and the second path is that enzymes that are in the synaptic cleft break down the neurotransmitters there, and then they'll recycle the material that it's made of.